Hello, everyone, and good morning. Thank you for joining me for another Precisionary webinar. And today we are really honored to host Dr. Balaji Krishnan, who is going to talk to us about phospholipase D signaling in health and disease. So Dr. Balaji Krishnan is really an accomplished associate professor. He is at the Mitchell Center for Neurodegenerative Diseases in the Department of Neurology at the University of Texas Medical Branch in Galveston in the United States. So visit him and you'll get some nice warm weather. Dr. Krishnan is deeply involved in exploring the molecular intricacies that underlie synaptic resilience in neurodegenerative states such as Alzheimer's disease and related dementias, such as frontotemporal dementia, or FTD. His expertise spans a wide range of research methodologies, from molecular biology to electrophysiology and behavior analysis, using both human post-mortem brain tissues and translational animal models. So in today's webinar, which is Phospholipase D Signalosome in Health and Disease, Dr. Krishnan is going to dive into multimodal assessments and biomedical research, talking about the importance of appropriate translational models and the crucial role of synaptic function in defining neurological health and disease states and neuropsychiatric and neurodegenerative conditions. So thank you for joining us today to gain valuable insights into the cutting edge work by Dr. Balaji Krishnan, and also finding out its impact on the field of neurodegenerative research. So for everyone, I just want to let you know that you are free to ask questions and make comments in the chat box, and I'll moderate it. And at the end of Dr. Krishnan's webinar talk, I'll open up the floor for an active Q&A session. Thank you so much. And with that, Dr. Krishnan, over to you.
Thank you, Abby. That was a very generous introduction. I appreciate it. Thank you all for joining. So today I'm going to be talking about some of the earlier research that led to us looking at why phospholipase D might be important in both health and disease. And I'll show you some of the published work at this point of time, and hopefully you will be able to gauge a sense of why it is important to look at different translational models and different approaches, including human clinical significance, when looking at neurological disorders. So without further ado, so let me go ahead and go to the structure of my presentation, which is going to be providing you why it is important to study phospholipase D as a potential therapeutic target in Alzheimer's disease and related dementia. In this case, I'll be focusing quite a bit on what we have done. So I'll first show you why PLD, because it affects memory mechanisms in the hippocampus and the amygdala. And we started off looking at the amygdala, and therefore I'm going to give you a little bit of a synopsis of the studies that were done to establish why looking at PLD1 was important in the first place. Then by looking at the different kinds of memory, how we can use multimodal assessments to corroborate your own results and make sure that you are proceeding in the right direction. So this is the structure of the amygdala, and this is a coronal rat brain slice. And at this point of time, when were using rat brains, these were young adults, if you will, 3 to 4 weeks, so not really that much of an adult, but it was amenable to looking at amygdala. So here is a coronal section from Paxinos and Watson. And what you can see is you can clearly differentiate, of course, the hippocampal area. But what is important for me at this point of time is to demonstrate to you that this particular structure, which is a nuclei of which is a structure made up of different nuclei, play a very important role in what is known as the emotional memory, and also it is the part of the dual amygdala hippocampus related associated memory. Therefore, by studying this particular structure, what are we trying to establish? That is what I would like to if you allow me to elaborate on it, provide you some insight on. So, as I pointed out, the amygdala synaptic pathway has been very well mapped out for fear conditioning, and this is from a review by Ledoux, who recently retired, by the way, from New York University. So here you have the auditory system shown here in green, and the somatosensory system shown here in red. Gray, and amygdala shown here in pink. What you have to appreciate is that inputs and outputs come from the thalamus via the cortex, and thalamus via the cortex for both these input. So if you have sound, which is the conditioned stimulus, coming into the lateral amygdala, that is then the information then goes over to the central amygdala and the basolateral amygdala and is modulated through the intercalated cells before it is transferred to the medial or goes over to emotional response. So what I want you to appreciate here, and this is the layout of the structure from a slice that was done, and you can appreciate how these structures are laid out. And here is the ITCs. This is the lateral amygdala. This is the basolateral amygdala, and this is the central amygdala. So you have inputs going this way, inputs going in these directions, and from here to here. So what you can appreciate here is, unlike what many of you might be familiar with the hippocampal circuits, there is an intermingling of information that happens here because of the nature of this limbic structure. So what we assessed initially was this particular response. So if you have a conditioned stimulus, which is a sound or a location cue, you pair it with an unconditioned stimulus, which in this case is aversive like shock, or cocaine, which is appetitive. So we use cocaine as an appetitive response because you can get the animals addicted to it, which means they like it. So that would be appetitive versus aversive memory. So I want you to think of it in that context more than addiction. So what it gives rise to is an unconditioned stimulus with the shock or cocaine will give you an unconditioned response, which is startle, preference, or hyperactivity. So you either have them startling in the chamber, or they'll show a preference for going into the chamber, or they will demonstrate hyperactivity in that chamber. So you can appreciate here that there are different kinds of ways in which you can look at the response. So then when you pair it with the CS, so you give the unpaired stimulus and then you pair it with a sound or a location cue where you place it and so on, you get what is called as a conditioned response, like a standard Pavlovian response. So then you can remove the unconditioned stimulus and you will still get the conditioned response if you place the animal back. And that is a sign of classical conditioning and associative memory mechanisms. So this was what was done using the rat model. As you can see here, it is kept in this startle chamber. So this has a piezoelectric device here. It has a sound mechanism and it is placed on a grid. And this is a 4 to 6 week old Sprague Dolly rat, male rat at that point of time. And you had three groups. One is the fear conditioned group, which received the pairing of the shock and the sound. The experimental group, which is also known as the paired or the FCFC or the paired experimental group, and the unpaired group, where they get the shock and the sound, but they are not paired. They are not within 2 seconds of each other. So this is the structure that was there: 10 tones and shock with those. And for the unpaired, it was 10 different CS and US, which was given in a pseudo random manner. So during the testing, the only thing that was played was the sound or the noise. So there was a white noise and it was interspersed with the sound. And that would be done in this particular chamber because this is a different chamber. So this would also allow you to appreciate the amygdala based response in this case. And what you can observe here very clearly is the unpaired animal shown here in green, the startle amplitude is very low, but in the paired group of animals, the startle amplitude is significantly higher. So using this particular approach, our group at that point of time, which was Dr. Gallagher, Shinnick Gallagher, with whom I worked as a postdoctoral fellow, we were looking for a signaling pathway. So at that time, what was established due to the seminal work from her lab in amygdala, she was a leader there, and was that NMDA circuits and receptor proteins are downregulated in the amygdala during maintenance of fear memory. So here is the difference between hippocampus and amygdala. Hippocampus does not store memory for the most part. There is some emerging evidence now recently that it does, but it basically acts like your, freeway, if you will, which is basically that information goes through it and it is transported to different places, but nothing resides per se in the hippocampus. The amygdala is not such a structure. Being more primitive, it stores the memory. So what happens is, in addition to getting the circuits modulated, you have biochemical modifications. For example, like there is a downregulation of certain NMDA currents, which is basically the excitatory pathway. So this happens in the lateral thalamo-lateral synapses. As I showed you, those are reduced. So here I'm giving you a pictorial representation of the state of the lab when I arrived there. And then again, internuclear synapse in the amygdala is not facilitated. So you basically do not have any activation that goes on here, no facilitation. And lastly, they exhibit different mechanisms of long-term potentiation. So one is L voltage gated calcium channel dependent and independent kinds of responses that goes from basolateral to the central amygdala. So suffice to say that there is a complex level of glutamatergic regulation that happens here. So we were very interested to see at that point of time what are the converging elements that are working here. So what we need to take into account is that at this point of time we were interested in the dopaminergic, which is the motivation based, since we were looking at both fear as well as appetitive conditioning, and also the glutamatergic, which is basically the excitatory pathway that gets excited and activated in the amygdala. So we were looking for something that would converge downstream of these receptors. So that is where there was a two-dimensional cell electrophoresis done to identify different proteins that are upregulated or downregulated in these group. And what was observed was one of these 286 proteins was aldolase A. Yes, aldolase A is definitely one of the most common enzymes that you find in all glycolytic pathways. But what is happening here, in particular, in this learned or memory associated pathway? So that's what we wanted to explore. And so, as you can appreciate here, compared to the unpaired, in the paired group, it is lowered. And we were able to show that the density is also lowered in here. So this is the normalized volume, as you can see from the 2D gel electrophoresis, and also straight Western looking at aldolase A levels. Now, aldolase A physically associates with phospholipase D. This is where the connection to phospholipase D came in the first place, because we knew aldolase A is a biomarker, so it can be mapped. But what is it actually associating with? And Kim et al. In 2002 were the first ones to show that at least in the neurons, this was playing a big role. And the aldolase A physically associating with phospholipase D gave us an indication that if this was low, then it was going to be affecting the levels of phospholipase D. So we looked at those levels, and what we observed was uh, over here, what we can see is that in the naive amygdala, there is very little levels of PLD compared to aldolase A. Okay, so what is phospholipase D? So let's get to the basics of this. So phospholipase D, or in this case, as represented as phospholipase D1, is one of the two major isoforms of a lipolytic enzyme that was first identified based on these HKD domains that are phosphatidylcholine catalytic domains. So this particular one that you see is 1074 amino acids, and it has a protein kinase C domain, a Rho domain. So it's also acting as a protein-protein interaction domain, and there is a loop structure that allows it to become independent. Now, it is usually present in the endosomes like the Golgi or the ER, but what happens is upon activation, it is hypothesized to come onto the membrane and regulate the curvature of the membrane. So you can appreciate that if it is regulating the curvature of the membrane in terms of synapses, it can regulate presynaptic, postsynaptic, as well as the actual dendritic spine integrity, formation, breakdown of dendritic spines, all of these things which are important for memory. So this was one of the avenues that I chose to explore. And so to give you an idea, to sum it up, because it was a lot of work, I just want to tell you, using these approaches, what we were able to show was if you take the dopamine receptors and the glutamate receptors located on the membrane in the amygdala synapses, both PLD isoforms, both known PLD isoforms that have a role for breaking down phosphatidylcholine play an important role in motivation and memory processes. So if you look at the control mice, what you can see is PLD1 and PLD2, as shown here in these two frames, associate with dopamine and metabotropic glutamate receptors, converge to give you memory and retrieval. Okay, so this is the normal status of what happens. So this recruited PLD1 stays there. In the pathological states, which is basically fear memory or in addiction or repetitive memory. Okay, and looking at pathological allows you to appreciate what can go wrong, right? So what you see here is that first of all there is increase in the cytoplasmic levels that we registered using Western blots, and then look using electrophysiology and molecular biology, we were able to show that there is increased recruitment of PLD1 and PLD2 to the receptors in this case, which results in a reinforced memory and retrieval, such that even days after the stimulus has passed, you can still see these changes being monitored. And this became the part of different papers. So, in addition to showing that dopamine and glutamate are involved, we were also able to demonstrate that serotonin was involved. So, PLD1 and 2 become these common convergent downstream elements that are important for memory formation and retrieval. So, this opens up a Pandora's box saying what happens if you remove PLD1 or if you overexpress PLD1 or if there is a perturbation in the homeostatic levels, as you will. Okay, so if you use electrophysiology lingo, that's another important thing that we were interested in looking at. So if you see here, what you can appreciate is reducing PLD1, as was done by several groups, three different independent groups that I'm aware of. If you generate a PLD1 knockout mice, okay, and I'm going to focus on PLD1 for the rest of the talk for obvious reasons, as it will become evident. Even though initially we found that both PLD1 and PLD2 are important in healthy states, in pathological states, the story is a little bit different. So here what you can see is that PLD1 mice are viable and grossly normal. There is some developmental defects that are associated, especially in the formation and so on, but it does not compromise the survival of the mouse. Yes, a little bit of social anxiety, social deficit, behavioral deficits were observed, but they were not detrimental to survival. So let me stress on that part. Detrimental to survival is the important part. Now, there are other aspects, like, for example, they confer certain kinds of protections, such as preventing metastasis by the action on thrombosis. And we'll come to that in a second. So, the other thing that became very prominent is recently there were three papers that came out looking at what happens if you have compromised function of PLD1, which means you don't knock out PLD1, but you have it compromised. And this becomes very relevant because PLD is conserved all the way from bacteria to humans. And it's also there in plants, okay? So there are different variations, which means that this is a very critical protein. Also means that it is duplicated and has redundancy of function. So by knocking it out, we are not necessarily going to eliminate the essential functions associated with it, especially in this case, but it is taken over by something else. There is compromised function. And how do we know this? Because if you look at these papers, and the one that I'm demonstrating here is by Larucci et al., what they showed was there is in these group of Japanese couple, they had four different children, and all four children had a homozygous copy of a reduced functioning PLD1 variant. Okay, so while the as carriers, the parents were not affected in terms of their day-to-day life, obviously, but their their fetuses, one of their fetus passed away in utero, and the other three made it but did not completely survive to even beyond a few days, showing that it is very important. And when they looked at post-mortem, it was chiefly because the right side cardiac wall had defects. So there was a specific area, and this we had known even when we're looking at PLD1. We used to look for the expression in the heart, knowing that the function of PLD1 in the heart plays a bigger role. So, in terms of survival critical role, reducing the PLD1 during development, not post developmentally, during development causes problem. However, the bigger problem is if you have post developmental increase in PLD1 levels. One of the things is in 1998, a paper, and the only paper that I've seen, shows that PLD1 expression is regulated by its own splice variant, a 50 kilodalton protein. But that, if it is lost or for some reason you see this PLD1 overexpression that occurs, you can see it being manifested in viral infection, predominantly in cancer, as well as some neurological deficits before, such as Crepey and so on. So there has been a lot of interest, especially from the group in Dr. Craig Lindsley, who's spearheading it, along with Dr. Alex Brown from Vanderbilt, in making small molecule inhibitors, particularly targeting cancer. Why? Because if you affect PLD1 post developmentally, as I showed you, it does not have much of a defect, but only the overexpression. So you can regulate these levels. So my question at this point of time was, is it relevant for memory loss in neurodegenerative states? So why neurodegenerative states? Because as all of us are aware, one way or another, Alzheimer's disease is the sixth leading cause of death in the United States. Between 2008 and 2018, deaths from heart disease have decreased, while deaths from Alzheimer's disease have increased by 146%, and one in three seniors dies with Alzheimer's or another dementia. It kills more than breast cancer and prostate cancer combined. So there is national and international recognition on the importance of this disease. So where am I coming here with this? I'm coming here with the basis that Alzheimer's disease has a synaptic basis, because that is where I am interested in it. What is the synaptic basis? So if you look at the two known neuropathological markers, okay, I know this is a very busy slide, so I apologize for it, but there is a lot of information that is known about this, okay? But what I want you to appreciate from this busy slide is there are many components that can result in aberrant synaptic transmission and impact cognitive function. And this can be because of effects in the presynaptic and in the postsynaptic terminals. Okay, so like for example, as you can see here, A beta shown here in these small reddish pink, if you will, I mean, oligomers. It basically affects everything from AMPARs to mGluR5 to efferins to SOL and so on, as well as NMDA. And tau, of course, has an effect from here going on to the synaptic vesicle cycle. So this is some of the aspects that are being explored and affecting PSD95, fin kinase, tau, resulting in loss of spines, causing excitotoxicity. Many of these features that I experienced to be common in looking at the role for PLD1 in long-term memory. So the question was how to look at this. So why are oligomers critical for studying the neurodegenerative states? So this is one of the things that we need to appreciate. So for being able to model it. So one of the essential things that we have to understand about Alzheimer's research is how to go about looking at it. There is, as I'm sure you have heard from news media as well as literature, there is a lot of effort in a lot of directions. Okay, while the success is seen at the level of translational models, there are a lot more problems that come when applying these immunoquenching therapies that are there in humans. Now, to complement this, there need to be ways in which we can look at it, and that is where I would like to direct your attention to why we are looking at it. So let's go back to the reductionist approach and then come back to the bigger picture. So oligomers are critical for studying neurodegenerative diseases because you can look at their toxic effects, okay? And this is very well established, so I'm not going to spend a lot of time. But these oligomers, suffice to say, are not the ones that form amorphous aggregates, but rather they remain in circulation and give rise to different kinds of problems, okay, including promoting region to region cells, I mean, spreading, cell to cell spreading, which is one of the theories that is there, as well as causing toxicity by actually physically affecting the synapses. And this can be very well demonstrated uh, in electrophysiology, as I'll show you in a minute. So PLD1 levels are elevated by A beta oligomers. And this is one of the first proofs that I'll show you. So here is a picture of a mouse from which we remove the brains and then we cut it. And this is where the importance of precisionary instruments comes in. No, I'm not trying to promote them, but it was very instrumental, as you can appreciate, when we are doing Alzheimer's disease, where we have to look at older animals, that you cannot really use a sucrose based cutting approach, but you have to use an approach that allows you to keep the synaptic integrity of aging animals intact. So, in addition to using Jonathan Ting's procedures of NMDG or HEPIs or other aspects that allow you to get a more robust recording. Precisionary provides this compressome, which completely encloses your compromised brain, if you will, with agarose, allowing you to slice it much better. So the integrity of the slices is much, much better. And we have been successful in going all the way up to 24 and even 32-month-old wild type mice as well as 3-XTGAD mice. Unfortunately, I'll not be able to show those data here, the 24 and 32, but the others I'll be able to show. Nevertheless, what we were able to appreciate was when we made these slices and we looked at the levels of PLD1, you can see that PLD1 shows up at 120 kilodaltons using the antibody. And what you can appreciate is that, okay, this is the problem of making the slides in Mac. So my apologies here. So what you can appreciate here is if you add the A beta oligomers, if you just add the A beta oligomers over here, so take my word for it, I don't have the graph here, but you can see that there is appreciable levels of PLD1 that occur. And this is within 1 hour. And remember, this is recruitment of the PLD1 to the membrane. And so this is what you see to be increased when you add PLD1, when you add A beta oligomers. Now, the PLD1 inhibitor, which I'll talk about, a small molecule inhibitor, if you incubate that along with the A beta oligomers, you can clearly appreciate that this particular increase in membrane association goes down. Okay, so having shown that there is definitely a change in the phosphorylation status of PLD1 as well as the absolute level of PLD1 recruitment to the membrane, there is a scientific rationale in exploring whether such elevated PLD1 is beneficial or detrimental to synaptic plasticity. So here we come to slice electrophysiology studies. This is I borrowed this from the Jove paper, and this is a method that was very well described by Dr. Krishnan and his group in about how to make slices and go ahead with looking at electrophysiological response, the gold standard when it comes down to looking at synaptic plasticity. And one of the easiest way for us to look at whether there are presynaptic, postsynaptic aspects to PLD1. So unfortunately, I'll not be able to show some of the presynaptic, postsynaptic. I summarized all those studies, but when we were doing amygdala research, there was components of PLD1 that was associated with the presynaptic and the postsynaptic component. But suffice to say that doing good electrophysiology will be very essential here. So, without further ado, basically this tells you that we took these sagittal sections or more horizontal sections where we were able to map the Schaefer collateral, and most of the recordings that I'll show you are with these Schaefer collaterals, although we have the capacity to do both mossy fiber as well as perforant pathway, which at this point of time we are still studying, and hopefully in a seminar down the line I'll be able to give you more results along those lines. But what we do is we stimulate on the area coming from the CA3 towards the CA1 region, and we record in the CA1 Schaefer collateral synapses, and that gives rise to these nice fiber volley based excitatory postsynaptic potentials that you can look at the slope for at the 20 and the 80%, as you can clearly observe here. And this gives us a metric. If the slope changes, you can appreciate the effect of the oligomer. And I'll show that to you in a little bit of detail. And again, like I mentioned earlier, this slice was made using the Precisionary instrument base. So at that point of time, and even now, it is one of the most reliable ways for us to get reproducible slices. Because of the compressing, you do not have the shearing of the tissue that happens in other cases where, if you use young tissue, it's fine. But for older tissue, the compressome really works very well. So what you see here is we make 300 micron sections. They are incubated with A beta or tau oligomers, and then we conduct the field electrophysiology. So I'm going to show you some of these traces. So to align you to the particular approach, here is the FEPSP slope in terms of percentage of baseline. And you can see that we do a 10-minute baseline followed by high frequency stimulation, which is the stimulation of 100 hertz. So now we give rise to what is called as what is commonly referred to in the field as cellular memory. So the Schaefer collateral synapses now show an increased response to the same stimulus. And you can appreciate this as you see that there is a 400% stimulation in this particular set of animals that we saw. This is again the control animal. And what you can appreciate is that it gives you a long-term potentiation that if you record for an hour's time, it's still maintained. And this last 10 minutes becomes important here. So now what happens if you apply A beta oligomers at 20 nanomolar? As I showed you before, it is incubated along with the ACSF for an hour, and then we observe and look at what the effect is. In terms of slope, it is slightly compromised, but not very different if you look summatively in the control. Now, when you apply 200 nanomolar, about tenfold more, you can clearly appreciate the decrease in the response. Okay. And if you put 2 micromolar, so if you increase it another tenfold, you are able to appreciate that there is a complete dampening of the potentiation. So this is a clear metric that you can use, a functional response of the compromised activity of the neurons as a direct readout for the effect of A beta oligomers. So you will recollect that I told you that when we add the A beta oligomers, PLD1 level goes up, right? So we want to see whether PLD1 is beneficial or detrimental. So how do you look at it? So you take a concentration that is intermediate and you ask the question, if you put the A beta oligomers along with the PLD1 inhibitor, do we see this particular kind of detriment, which means that PLD1 is protective, or we see an increase, okay, of the response, which means that inhibiting PLD1 prevents the activity of A beta oligomer toxicity of the synapses. So that means PLD1 increasing levels are detrimental. In other words, A beta recruitment of PLD1 makes it bad. Okay. So let's see what happens when we choose this particular response. Okay. And for the rest of the things, we will look at these long-term potentiation because over here we have post-tetanic potentiation and so on. But the recording stabilize at last 10 minutes. So you can take an average of this. And when you plot it, you are able to appreciate the changes. So the same representation that I showed you compared to the control, 20 nanomolar A beta, 200 nanomolar A beta oligomer, and 2 micromolar A beta oligomer show a decreasing trend, as you can appreciate. Okay. So now if you take the tau oligomers, you can demonstrate a similar profile. And some of these studies are earlier done by Dr. Octavio Arancio at Columbia University. And inspired by his abilities, we looked at these different concentration of tau oligomers. And the oligomers were kindly provided by Dr. Rakesh Kayed here in the Mitchell Center. And we at Mitchell Center, we have a very collaborative group. So we basically interact with each other very well. So what you can see here is if you take 5 nanomolar, similar to what you see with the A beta, you can see that there is a change. There is no change at 5 nanomolar, but at 50 nanomolar, you can see a decrease. And at 500 nanomolar, you see a complete loss of the potentiation. So here I demonstrated to you both A beta and tau oligomer independently having an effect on the synaptic neurotransmission at the Schaefer collateral synapses. So if you again look at the last 10 minutes, you can see this graded profile happening for the tau oligomer application. So again, coming back to the question, if PLD1 isoforms expression is altered in human brain, then there is a clinical significance. So even before proceeding to look at what PLD1 looks like, our group wanted to establish the human clinical relevance. And having Dr. Taglialatella on as our director for the Mitchell Center, I had access to some of the brain regions, so I could start looking at different aspects. So here, if you look at A beta plaques, you can classify them into stage 1, 2, stage 3, 4, and stage 5, 6, marking the stage of progress, which is better represented with the neurofibrillary tangles, thanks to Dr. Clifford Jack's work and their collaborators' works and all many others. You can clearly see the classification of A beta and tau distribution. So when we look at different aspects, okay, we looked at some of these responses. And what I'm going to show you here, I mean, this is published, but what I wanted to show you was when you look at the human clinical significance by taking the control versus the AD brains, the hippocampus in particular, what you see is that you have increased levels of PLD1 in the AD brains. So this corroborates with what we saw with the A beta oligomers. So when we are able to show that, we are also able to see that there is actually increased levels of PLD1 in the AD hippocampus, especially in the P2 fractions. Now, going further, we also demonstrated that if you look at the PLD1 colocalization A beta using the 4G8 antibody or tau 5, you are able to appreciate that compared to control, there is increased expression of PLD1 as seen by these PLD1 structures that you see here. Okay, both in these both cases of the AD plates that you see here. Not only that, when we looked at the profile in 6 different individuals, you can clearly appreciate there is increased co-localization of APLD1 A beta and PLD1 tau in these particular patients, subjects compared to control. So this allows us to establish that there is rationale behind pursuing this pathway. Now, there were 217 clinical trials in Cochrane database that confirm that reducing the activity of the PLD1 enzymes by increasing the substrate, which is lecithin containing phosphatidylcholine, results in temporary memory improvement. So all these factors add together to say that if we look at attenuation of PLD1 as a therapeutic response, it may have benefits. So how do we demonstrate that? So this is where we come back again and ask the same question. This is the question that we ask. Is PLD1 sufficient for A beta oligomer driven synaptic deficits? And what you can appreciate here is we took the 200 nanomolar A beta oligomer, and here is a representation shown in here in red for your convenience. Now, if you add a 2 nanomolar of a PLD1 inhibitor called VUO155069, again generated by Dr. Lindsley and Dr. Brown, so this, if you add it at 2 nanomolar, what we are able to see is that is not much difference, as you can see, in terms of improving any kind of response. So 2 nanomolar was 30 down. And now if you take 20 nanomolar, as is seen by this inverted triangle, you see that the response goes up a little bit, saying that if you inhibit PLD1, you are able to prevent the detrimental effect of A beta oligomers. If you further use 200 nanomolars, you are seeing that it's overlapping with what we saw with 20 nanomolars. So, in other words, you are seeing a definitive improvement compared to the A beta oligomer treated slices. So, PLD1, the inducible isoform, therefore, is recruited by A beta oligomers for the synaptic dysfunction related aspects of Alzheimer's disease. Okay? So, what about PLD2, which is the other known constitutive isoform? Okay, so now it has structural similarity as well as is constitutively present and has been demonstrated by another group that if you take PLD2 knockout mice along with PG2576, you can prevent Alzheimer's disease. And this was shown in 2010 by Gilberto Pavlo's group at that time in Colombia. So we looked at PLD2 specific inhibitors. And what we were able to appreciate was this is VU0364739. Now there are better ones, but this one was the original one we used. At 2 nanomolar, again, to the A beta oligomers, we did not see much of a difference. But at 20 nanomolars, it went up and the response improved. And at 200 nanomolar, it was much better than what we saw with PLD1. So therefore, there is definitive corroboration to what Gilberto Pavlo's group showed that PLD2 definitely has a role in A beta oligomer related synaptic dysfunction. So this is also recruited by A beta oligomers. So what is the importance of looking at PLD1 over PLD2? And let me come to that in a second. So this is summing up what was published where we showed that there is a concentration dependent improvement of response, okay, for PLD1. So suffice to say what I showed you earlier is better indicated over there compared to this one. So coming back to the tau oligomers. So that's where we are looking at it. As you can very well appreciate, A beta oligomers are the initial cohort that basically keep on increasing, and then tau gets recruited by A beta oligomers according to one particular hypothesis. And of course, tau is known to be present in 22 other neurodegenerative states as a toxic form in this, causing more destruction. So, in recruitment of tau, is also an important aspect. So we wanted to see whether tau is affected. So we took tau oligomers, and like I showed you previously, we looked at whether the PLD1 inhibitor is effective against tau oligomer-based synaptic dysfunction. And what you can appreciate here is, yes, it is. So PLD1 inhibitor shows at 2 nanomolar did not have a difference, but at 20 and 200 nanomolar definitely showed improvement. What about PLD2? Well, when we looked at PLD2, okay, I don't have it here. Okay. So PLD2 inhibitor, I don't have the profile, but I can show it to you over here. You can see that compared to this particular tau oligomer dependent blocking of the synaptic response, it does not improve. Application of the PLD2 inhibitor at increasing concentration did not have an effect on tau oligomers. So it seems like at least earlier, the constitutive PLD2 is affected by the A beta and A beta effects. But then, as PLD2 gets depleted or affected, I'm not sure what exactly happens, but that's something that needs to be tracked. PLD1 now becomes the central player and is recruited by both tau and A beta. And so now it carries on the synaptic dysfunction. So can we treat it? So that was the next step. So the therapeutic aspect or estimation of this therapeutic potential was important. How did we do this? First of all, we established the effect on cellular memory mechanisms. So what I showed you here, you are able to appreciate. If you look at the synaptic neurotransmission, we were able to show improvement for both A beta and tau driven synaptic dysfunction. Now, does that translate into improvement of memory? So, if you take your regular wild type mouse at a younger age and you subject it to A beta oligomers ICV or tau oligomers ICV, and then you give the VUO1, what we were able to see was it improved the novel object recognition. So A beta oligomer drops the object discrimination index to 50%, which means the animal now fails to recognize the new object compared to the old object, whereas the control shows a higher propensity to spend more time with the new object. When you apply VUO1 by itself, it doesn't have an effect. But when you apply A beta oligomers along the VUO1, it improves the response, which means that by blocking or attenuating, if you will, PLD1 activity, you are able to prevent the A beta oligomer related synaptic dysfunction and underlying memory deficit, which includes novel object recognition. The same thing we were able to observe with the tau oligomer. So this gave us preclinical validation to pursue PLD1 inhibition as a potential therapeutic target. As I already showed you, there is human clinical relevance, and now I showed you mouse or preclinical model-based approach. Now, is it possible for us to look at progressive accumulation? Because that is a very important factor when you consider Alzheimer's disease, right? When you're looking at Alzheimer's disease, it is progressive in in addition of these oligomers and oligomer-based effects that results in the neurodegenerative profile that you see, which is one of the things that was not being prevented. So we looked at a particular model, and a reproducible model that expressed both A beta and tau is the 3xCG-AD model developed by Dr. Laferla's group in 2003 and reported in 2003. So what happens here, as you can see, is you can see cognitive impairment around 3 months of age. This is from the forum. And at 6 months, you are able to appreciate plaques. You're able to appreciate changes in long-term potentiation and long-term depression, as well as gliosis. And as the animal progresses to 12 months, you start seeing tangles also. So this is a very good model for us to look at these profiles. So we then ask the questions, do we see any kind of PLD1 association? So what I am going to show you here is that at 3 months where there are plaques, at 6 months where there are plaques, you are able to see an expression of PLD1. I'm not comparing it to the wild type because we want to see whether it progressively increases. So if you look at 12 months following this, which is where you start seeing both A beta and tau, you can see that there is increased levels of PLD1. So this maps with what we showed earlier, where there is recruitment of PLD1 by both A beta and tau towards the synaptic dysfunction. Okay. And then lastly, if you look at 18 months, which is a bigger, I mean, a more progressive profile, you can clearly appreciate the significant increase in the PLD1 expression at the P2 synapse. So this gives us enough rationale to use this as a transgenic model, the 3-HTG-AD, to study AD-like cognitive decline plus the potential therapeutic effects. Now, here comes the bigger issue, which is how do we go about it? So, what becomes important here is not to use just one way of assessing things, but use multiple ways of assessing things functionally. So what we did here was, again, as you will appreciate, I am interested in looking at the amygdala also. So I put in one more behavior, and let me come to that. So what I'm going to show you here is, if you take the 3-XTG-AD mouse, age it to up to 5 months, okay, and then you give it an injection intraperitoneally of saline at 0.9%, or the VUO1 at 1 mg per kg IP every other day for a month, then we can, look at multimodal functional studies, which includes novel object recognition, fear conditioning, followed by electrophysiology, and then look at whether there are changes that we can map at a group level, which is basically Golgi-Cox staining to look at dendritic spine integrity, which is one of the mechanisms that I'm going to talk about today. There are others, but I won't have time to talk about it today. The other one is to age. So this is where you are going to have a lot more A beta plaques at between 5 and 6 months, right? Between 11 and 12 months is where we see both A beta and tau effects. So we aged another cohort from 11 up to 11 months, and then uh, provided the same kind of treatment at the same concentration to see early and late stage effects of the VUO1. And what I show you here is that if you first of all look at what happens, the immunohistochemical profile of PLD1 levels because of this administration, and I'm giving you a demonstration here at 12 months, you can clearly appreciate PLD1, which is shown in red here, in the saline treated sibling cohort is increased in the DG region compared to what you see with the inhibitor. Okay, so it is reduced, and you can see the demonstration here. Not only is it reduced, of course, this one says tau, but this is A beta, I'm sorry. If you look at the integrated density of PLD1 and A beta, which is the co-localization, you also see that that is diminished. So both A beta and tau are diminished, or their association with the PLD1 is diminished by treatment with the PLD1 inhibitor. So having shown you this particular part, then I want to show you what happened when we look at it in the early stage. What we were able to appreciate in the novel object recognition is that the object discrimination index improves in the inhibitor treated group, shown here in green, compared to its sibling cohort. Similarly, we were able to see that the contextual fear freezing was much better and improved here, although the cued was not, which is the amygdala based one. But that is a discussion for a separate time. Just because of time, I'm going to have to stop shortly. But the fear conditioning we were able to corroborate what we saw with novel object recognition. And I want to emphasize that such kind of approaches are important when we are looking at translational studies, because we want to verify that a phenomena has a good underlying basis. And so here I want to emphasize that this doing this particular object discrimination index in novel object recognition followed by fear conditioning was essential for us to corroborate what we are seeing to be an important outcome of the inhibitor treatment. Then, of course, we verified the improvement in synaptic function by looking at the Schaefer collateral synapses, like I showed you elaborately earlier. And then we were able to do Golgi staining, where we were able to see the number of mushroom spines being preserved in this particular group. So, in other words, by preventing the PLD1 enzymatic activity, we were able to demonstrate at least that one of the changes that we saw was in the integrity of the mushroom spines. So there was preservation, or there was some amount of synaptic resilience conferred because of preserving these dendritic spines due to reducing the activity of PLD1, which is a normal outcome. Now, extending it further, and of course, this is the crew that actually did the work here, and I'll acknowledge them down. So Here again, this is the same profile, but now done in the 12-month-old 3xTG. So you have to appreciate that when these animals undergo so much of behavior and so on, you want to maintain the integrity. So having a good slicing mechanism and looking at electrophysiology is going to be very important. So we took things a little bit further here. We looked at low frequency stimulation, LTD, or long-term depression, in addition to frequency stimulation, LTP. And this was important, I mean, maintaining everything else the same, because LTD will give us a view of what happens with the tauopathy. And tau is known to affect LTD also. And of course, presynaptic, but in this case, we were not able to see any pad pulse differences, and we'll come to that. We won't actually come to that today. But what I would like to demonstrate is that the improvement that you see in both the long-term depression shown here in green and the long-term potentiation, okay, that was again replicated by improvement in the novel object recognition and fear conditioning, both contextual as well as cued. And again, over here, we did not observe an improvement in the mushroom spine preservation, but instead we saw that there is an increase in the filamentous spine length. So it is important to appreciate that these are older animals, and we started the treatment at 11 months. So even demonstrating this particular change was important for us to show that there is a variation that we observe. So what I want to leave you with, again, this is the same representation to show you, similar to the other format, but I thought this was more informative since you are able to see the long-term potentiation and long-term depression and look at the actual data. Okay, so to summarize, because of time, I have to summarize here. There is a whole lot of data that people in the lab are generating, but hopefully at another forum we can discuss that a little bit more. But PLD signalosome is important during development and in healthy animals. And we were able to demonstrate that it is involved in the formation of amygdala long-term memory. Okay, there we used rats to show, using behavior, electrophysiology, pharmacological approaches, and biochemical approaches, that it is a very significant factor that is involved with normal memory formation. However, if you have aberrant elevation of PLD1 or recruitment, if you will, it is detrimental to memory, okay, especially in the hippocampus, as we demonstrated using these approaches, where the integrity of the dendritic spines are affected. And this is because of the activity of both tau, A beta, and tau. So we were able to use a reductionist approach to show you how A beta and tau are playing a major role in the synaptic dysfunction and memory deficit in these transgenic models, and looking at that specific effect of PLD1. Now, there is an attenuation of PLD1 using small molecule inhibitor by attenuating, not eliminating, attenuating it, both at early and late stages in the progression of the neurodegenerative state with A beta and tau in this late onset model. So 3xTG is very well established now as a late onset model, especially after the remarkable amount of work done by La Ferla's group recently, which they published, Javonilla et al. 2021. And they showed that it promotes resilience to the progression of cognitive decline. So we are able to say, at least using these preclinical models, both rats and mice, of how PLD is important. There are some more transgenic animals. I mean, my initial training was in Drosophila, so we are looking at some of these aspects. But I hope to be able to provide you with more of that. But nothing would be complete without acknowledging my team. And I am showing you some of the team here, but all of them are very hardworking. Chandramouli is a senior graduate student. He's graduating out. Shravan is a postdoctoral fellow. Sarah is a research technician. Clarissa is the research associate. These are medical students that came in. So this is Kyle, and this is Evan, and here is Joshua, and here is Helen. So we have a good population of medical students who are getting trained here, and Jared, without whom we could not be doing all of the slicing and so on. So thank you, and this is the funding that we received for doing some of these work. And with that, I would like to thank you and be happy to answer any questions.
Wonderful. Thank you so much, Dr. Krishnan. So I want to open up the floor to the individuals who are here and see if you had any questions or comments. Feel free to unmute yourself and show your video if you'd like. And we're just going to have a very friendly Q&A chat.
So I'm going to stop sharing at this point.
Okay. And of course, if you don't have any, that's okay too. So I'm just going to give it about 10 seconds to see if anybody's interested. And if not, then I'm going to thank everybody. And sometimes, Dr. Krishna, we get questions afterwards just by email, and we will send that to you and put you in touch with our attendees. So at this point, I just wanted to say thank you very much and wish everybody a wonderful week.
Thank you. Thank you very much for this chance. Take
Bye-bye.
care. Bye.